Maurice Wilkins, Rosalind Franklin, James Watson, and Francis Crick were the key contributors to the discovery of DNA structure. Franklin was a pioneering X-ray crystallographer whose work provided critical evidence for DNA structure. Her famous Photo 51, an X-ray diffraction image of DNA, showed its helical structure and dimensions. Maurice Wilkins was a British biophysicist whose X-ray diffraction studies of DNA proved crucial to the determination of DNA's molecular structure by James D. Watson and Francis Crick. James Watson, an American geneticist and biophysicist, along with British biophysicist Francis Crick, played a major role in the discovery of the molecular structure of DNA. In 1953, Watson and Crick proposed the double helix model of DNA. They used existing data, particularly from X-ray diffraction images, to suggest how the structure of DNA could carry genetic information. Watson and Crick, along with Maurice Wilkins, received the Nobel Prize in Physiology or Medicine in 1962. Watson and Crick model features: DNA has a double helix structure with two strands running in opposite directions, antiparallel. Sugar phosphate forms the backbone. The nitrogen base holds together the two strands through hydrogen bonding. Nucleotides are the basic building blocks of nucleic acids, such as DNA and RNA. Each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogenous base. Sugar can be ribose in RNA or deoxyribose in DNA. The nitrogenous bases in DNA are of four types, namely adenine A, thymine T, cytosine C, guanine G. The nitrogenous bases can be either purine or pyrimidine. Purine bases include adenine and guanine, while cytosine, thymine in DNA, and uracil in RNA are pyrimidine bases. According to Erwin Chargaff, who proposed Chargaff's rule, the amount of adenine A is usually similar to the amount of thymine T, and the amount of guanine G usually approximates the amount of cytosine C. Complementary base pairs are observed in DNA where adenine A pairs with thymine T via two hydrogen bonds. Cytosine C pairs with guanine G via three hydrogen bonds. Importance of complementary base pairing. During DNA replication, complementary base pairing ensures that each new DNA molecule is an exact copy of the original. Base pairing is critical in the processes of transcription, DNA to RNA, and translation, RNA to protein. The sequence of bases encodes genetic information, and complementary pairing allows for accurate reading and expression of this information. The hydrogen bonds between the bases contribute to the stability of the DNA double helix, with GC pairs being slightly stronger due to 3 hydrogen bonds compared to AT pairs. DNA can be classified into 3 types based on its structure. aDNA. It's a right-hand helical structure that is more compact than bDNA. Typically found under dehydrated conditions. Shorter and thicker than bDNA, and base pairs are tilted relative to the helix axis. bDNA. The most common form of DNA in living organisms. It has a right-hand helical structure. Base pairs are perpendicular to the helix axis and has about 10.5 base pairs per turn. zDNA. A left-handed helical structure that is less common. Formed in regions of DNA that are rich in GC content or under certain conditions like high salt concentration. Zigzag shape. With alternating purine and pyrimidine bases. An RNA nucleotide is composed of a ribose sugar, a phosphate group, and a nitrogenous base. RNA contains 4 nitrogenous bases, namely adenine A, uracil U, that replaces thymine found in DNA, cytosine C, guanine G. Types of RNA. Messenger RNA or mRNA. Carries genetic information from DNA to the ribosome for protein synthesis. Transfer RNA or tRNA. Transfers specific amino acids to the ribosome during protein synthesis, matching them to the mRNA code. Ribosomal RNA or rRNA. a component of ribosomes, essential for protein synthesis, helps catalyze the formation of peptide bonds. Other types include small nuclear RNA, snRNA involved in RNA splicing. MicroRNA, mRNA regulates gene expression. Long non-coding RNA, LNCRNA involved in various regulatory functions. Follow and subscribe us for more videos.